Good afternoon, everyone. Welcome to the ARC Training Centre in Predictive Breeding seminar series. Thanks a lot for joining us online and here in person. First of all, I would like to acknowledge the traditional owners and their custodianships of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. To introduce myself, I'm the director of the ARC Training Centre in Predictive Breeding, Professor Lee Hickey. It's a pleasure to be hosting this exciting seminar today. And before we start, just to remind you all, this seminar is being recorded. So hold your questions for the end. If you're online, please add your questions to the Q&A chat online so we can filter through those. So use the Q&A tab and we'll have a look at those questions at the end. We have 1 hour for the seminar session, and for those joining here in person, we've got some snacks and tea and coffee afterwards, so please hang around so we can catch up and interact. Today, it's my pleasure to introduce Professor Emma Mace. And to introduce Emma, her research interest is in developing and applying innovative genomics approaches to support sorghum improvement activities. In her current role, leading sorghum genomics research components of research projects funded by the GRDC and Bill and Melinda Gates Foundation, the ARC, her work focuses on generating significant innovative outcomes across a range of applications from basic through to applied, specifically in using technologies to bridge the gene to phenotype gap and to elucidate the genetic basis of quantitative and qualitative traits. And to recap, I would say that Professor Emma Mace is really a synonym for sorghum genetics and genomics, and she's a real world leader in this space. So it's a pleasure for me to introduce Emma. Please make her feel welcome.
Thank you very much, Lee. And I'd like to start by thanking the Training Centre for giving me the opportunity to talk to you today about panning for genetic gold. So today I'm going to talk to you about developing and deploying innovative genomics resources to support plant breeding. And in particular, I'm going to do so with reference to sorghum. Sorghum is the crop that I've worked with the most over the last 2 decades. It is a tropically adapted C4 grass species, the world's 5th most important cereal crop, which is a close relative of sugarcane, maize, and millet. It has a pretty small genome and high levels of genetic diversity for most traits, and is adapted to a wide variety of environments. So I created a bit of a tongue-in-cheek title for my talk today, comparing the process of developing new elite sorghum lines, which have beneficial sets of alleles and genes, to panning for genetic gold. Another phrase people often use in this context is finding the needle in a genetic haystack. But what plant breeders need to do is search through the equivalent of not just one haystack, but a whole farm full of haystacks in order to identify relevant genes from a broad range of material, including wild relatives, and stack them in suitable combinations of elite lines. With recent technology developments over the last decade, we now have a range of new omics approaches we can use to help us find valuable genes from a range of different material. So first of all, let's look at why do we need to use omics technologies? What's the challenge and how do we use them? Well, one of the major issues is that increasing the rate of genetic gain for complex quantitative traits remains pretty difficult in crop improvement programs. And why? Well, in order to breed for them, we need to understand the genetic factors and the interactions with the environment that underlie complex phenotypes, and to use this information to increase the response to selection. This response to selection can be influenced by four main elements as defined in the breeder's equation. And genomics can impact on all elements of the breeder's equation by helping to select for increased genetic diversity and to increase useful phenotypic variants, by increasing selection intensity, increasing the accuracy of the heritability measure, and decreasing the time required from making a cross until using an individual from that cross. However, genomics alone will not maximize the response to selection. Instead, what is required is a set of interlinked and complementary resources that can be used in multiple ways to support activities in breeding programs. Before I show you the resources, I'm going to use the same analogy about the people. Plant breeding is, by its nature, a multidisciplinary activity, including field and glasshouse work, phenotyping, genomics, data management and programming, statistics, quantitative genetics, simulation modeling, and of course, being anchored by the core disciplines of breeding. People do these roles. I do not work in isolation. This is a small jigsaw puzzle of some of the amazing team that I work with. And this is just the people that I'm co-located with in Warwick. And this includes both University of Queensland staff and DPI. The team extends well beyond Warwick, and of course, I'll acknowledge them at the end. The purpose of this slide, though, is to highlight the work I will be presenting is in large part not work that I've done myself. And I want to acknowledge and make sure that people understand I'm able to stand on their shoulders today and give this talk. So let's have a look at the resources that we've developed and use, which includes a range of genetic populations, data resources, software tools for use in crop improvement activities, which when used together offers opportunities to rapidly increase the response to selection. What I'll do now is briefly focus on individual elements, starting with genetic populations. So to make use of the diversity in the sorghum gene pool, we have developed a range of genetic resources that span the genetic variability available from wild and weedy through to highly elite germplasm. This includes a large diversity panel consisting of over 1,000 individuals from diverse geographies covering all of the major racial groups of sorghum, color-coded in the spinning PCA as the tips of the pyramid. And this is an excellent entry point to explore native variation in the species. We also have developed a large nested association mapping population consisting of both wild and diverse exotic lines crossed to an elite adapted line, with over 100 populations totaling over 5,000 progeny. The parental lines are the ones color-coded in this PCA, and you can see that it doesn't capture quite the extent of diversity of the diversity panel, but it provides the opportunity to look at these exotic alleles in the context of a genetic background adapted to the production area. We also have multiple elite populations from pre-breeding trials at advanced yield testing stages, color-coded here as blue and pink. And it gives you an idea of the more limited diversity of these elite lines in comparison to the other genetic populations. But what is particularly powerful is the interlinked nature of these resources and their complementary diversity levels, their resolution, that is how near we can get to a gene when we use them in mapping studies, their power and their suitability for performance testing in farmers' fields. So the diversity panel provides high resolution, but only low power, and is not so suitable for performance testing in farmers' fields due to large variability in height and maturity. whereas the NAM and the pre-breeding lines provide us with higher power and suitability for testing in farmers' fields, but the NAM provides slightly more resolution than the pre-breeding lines. So, depending on the situation and the trait in question, we can use these different populations in complementary ways to help us answer specific research questions. Yeah, just make that one. So let's now look at the data resources element, which, when combined with our population resources that we just looked at, allow us to identify genes underlying complex phenotypes through a P to G or a phenotype to genotype approach, as well as understand how functional gene changes impact on the phenotype through a genotype to phenotype approach. Let's have a look at some of these in more detail, starting with the sequencing resources and first the pangenome. To set the scene for this topic, I want to go back to one of my earlier slides, talking about the challenges of making genetic gain for complex quantitative traits. So one approach is to identify the genes underlying the complex phenotypes. And this raises the issue that until very recently, the majority of gene identification methods relied on the assumption that all genotypes contain the same number of genes. But do they? So to address this question, I'm going to show you two graphs. The first relates to the dramatic decrease in the cost of sequencing a whole genome, shown here in terms of the cost of sequencing a human genome. This dramatic decrease in cost has been driven by a wave of sequencing technology developments, enabling astonishing increases in throughput and accuracy. These platforms were initially largely used in a medical context, but very happily for plant scientists, reduction in costs have opened up new sequencing opportunities across hundreds of species. In fact, costs have reduced to the extent now that researchers have been able to sequence multiple individuals per species, as you can see here from the color coding in this graph. So considering the human genome, to illustrate this point, in 2003, the first human genome was published, and it was based primarily on the genome of one scientist involved in the project, Craig Venter. At that point, everything we knew about the human genome and the genes identified in it were based on Craig's DNA. It took almost 15 years to complete and cost over $1 billion. Now, scientists are on a path to sequence 1 million different human genomes at a fraction of the cost to get better capture the wide diversity of human populations in the world. The same issue exists in crops, in that the wealth of genetic variation available in all species means that a single individual selected to generate a whole genome sequence can never be representative of the diversity in the species overall. And this is exactly what has been found. By generating high-quality genomes of multiple individuals within a species, it has become increasingly clear that the use of a single reference genome per species can be very limited, and that pangenome analysis offers opportunities to overcome limitations of using a single reference genome through the investigation of the entire set of genes within a species. This figure here is taken from a recent publication where we're trying to illustrate that a single reference genome can only shine a light on a limited part of the entire gene pool. Whereas if you have multiple genomes, you can extend the part of the gene pool that you can see. So what is a pangenome? The pangenome represents the entire set of genes within a species, consisting of a core genome containing sequences shared by all individuals of the species and the dispensable genome. In other words, a pangenome refers to the universe of genome sequences existing in a species. It was first conceived 2 decades ago for bacteria and was first applied to plant species just over a decade ago. Since then, and with the evolution of a pretty large number with the evolution of multiple technologies, a pretty large number of plant genomes now have been developed. But to develop a pan-genome, still significant investment must be made. So we need to be clear on what we're getting. Why is a de novo genome helpful? So what can a pan-genome tell us that a single genome can't? Whereas a single genome can help us identify SNP variation between individuals, multiple genomes can more readily help us identify deletions and insertions between individuals. It can help us identify copy number variation, which can identify different duplication levels across genomes, as well as other types of structural variation, including inversions and translocations. So the group at Warwick created the first sorghum pan-genome around 5 years ago. All right, just go back. Our sorghum pan-genome was based on 16 carefully selected lines designed to try and capture the majority of variation within the primary gene pool of sorghum. The photo here is of Yongfu Tao, the postdoc who led this work. The spinning PCA is based on the diversity panel with the lines selected for the pan-genome color-coded. And as it spins around, you can just about see a colored dot in each of the peaks of the pyramid. We sequenced using a combination of Illumina and PacBio sequencing libraries, resulting in around 200x coverage per genome. I will say that in the years since we started this project, the technology and platforms have changed vastly. And now with the same amount of funding, we would be able to generate a lot more data. This is a very fast-moving area. One of the key interests with this data was to look at gene variation within the cultivated gene pool. We identified just over 60,000 gene families in total, with gene number per individual genome ranging from about 32,000 up to 37,000. So a fair amount of structural or presence-absence variation was clearly identified. We then wanted to look at how these gene families were distributed across the individual genomes and found that around 1/3 were identified as core genes. That is, they were present in the vast majority. About a further 1/3 was defined as shell. They were identified between 2 and 14 genomes, and that remaining 1/3 approximately was identified only in one of the genomes and identified as cloud genes. We can then explore the differences between these three classes of genes, and we can find that they have structural differences between the groups, with core genes being longer and with more exons than the other two types of genes, and with shell genes found to be more variable and having higher non-synonymous to synonymous substitution ratios than core genes, supporting their faster evolution. We were also able to explore the differences in gene function between these gene classes through comparing frequencies of GO, gene ontology term enrichment, and found that dispensable genes were significantly enriched with those terms involved in biological processes such as defense response, secondary metabolic process, and carbohydrate transport, whereas core genes were enriched for core biological processes, including reproduction, system development, seed development, and chloroplast organization. Overall, we were able to build up a more in-depth picture of the genome landscape of sorghum, shown here as sorghum's 10 chromosomes in a clockwise direction, that enabled us to identify features of the genome with higher proportions of genes affected by structural variation, and in particular found that genes in recombination elevated euchromatin regions, shown in green in lane A of this figure, are less likely to be affected by large effect mutations compared to the heterochromatic regions. and that we could identify higher levels of genetic diversity in these euchromatic regions in comparison to the heterochromatic regions, which gives us insights into driving forces creating genetic variation and purging deleterious mutations. So in summary, the development of the pan-genome within sorghum is allowing us to address a range of research questions, including insights into the genome dynamics between wild and cultivated sorghums, in addition to insights into structural variation in gene networks. Doreen Ware's group at Cold Spring Harbor have developed a centralized hub for the sorghum genomics community, Sorghum Base, to make our pan-genome data accessible through this site. We are now working on a new project in the ARC Center of Excellence for Plant Success to extend our sorghum pan-genome and generate a grass pan-genome. I've highlighted the additional species that we've targeted, that is maize, which diverged from sorghum around 10 million years ago, and rice, which diverged from sorghum around 50 million years ago. We're making use of pre-existing published genomes from maize and rice and integrating these with our genomes from the sorghum pan-genome. I wanted to show you an example here to demonstrate the benefits of comparative genomics. This was from a piece of research myself and David Jordan published over a decade ago. We compared output from our sorghum NAM on QTL, identified for maturity, with the results based on a comparable NAM resource in maize, where around 25 exotic lines were crossed to the same elite line. And that's the image you can see there. And what we found amazed us. Of the 75 regions associated with flowering time in maize, almost all of them, 90%, were nearby to a QTL in sorghum, and just over 1/3 were incredibly close. And what we can see from this example here is that for one chromosome in sorghum, and sorghum's chromosome is in the middle, many of the QTL mapped to 2 QTL in maize. We can think of maize as a tetraploid sorghum. But what I would like to point out is that in some instances, the maize QTL occurred in a region of reduced recombination, and the sorghum QTL occurred in a region of high recombination, and hence sorghum can provide greater resolution and more information for maize. So in this sense, maize and sorghum are providing mutual insight. I'm just going to show you 2 results from our work building a grass pan-genome. The first is a comparison of the number of genes per genome and the average per species, with maize being the most variable in terms of gene content, followed by sorghum and then onto rice. The next figure is a Venn diagram showing the number of genes in common and the number of genes unique per species, with just 17,000 genes in common across all 3 species and a range from 13 up to 26,000 unique to the individual species. So we are now interrogating these data to look at functions of the genes in common between the species, the feature and functions of genes unique to certain species, to provide us with more information to address questions such as, why is sorghum more drought tolerant than rice or maize? So the pan-genome projects that I've just described all rely on de novo sequence generation, and that's pretty expensive. A related but cheaper technology is called resequencing, and this allows us to look at whole genome sequence variation, mainly just SNPs, but on a much larger number of lines. So around 15 years ago, a group at UQ, together with David Jordan, Ian Godwin, and BGI, generated what at that time was the largest resequencing resource globally in sorghum. It was based on 47 very carefully selected lines with high confidence resequence data. And what we did is we used that data to look at patterns of diversity across all of the chromosomes and all of the genes in the genome, including chromosome by chromosome sequence diversity, the number of SNPs per chromosome, And over the last 10 years or so, we've mined these data at different scales to allow us to look at variation genome-wide or at any gene in the genome of interest. In the last 10 years, costs of resequencing have also dropped dramatically. And recently, we have increased our resequencing resource 50-fold and have resequenced not only the entire diversity panel, but other lines as well. And now, together with a range of colleagues from many organizations, we are mining this resource across a range of projects looking at fundamental and applied research questions. Another study we've been involved in is to extend the presence-absence variation dataset from our very limited pan-genome sample to the full diversity panel. And we've used read coverage of the resequencing data we have to determine gene-based presence-absence in the 1,000 lines in the diversity panel. And what we found was that from approximately 30,000 gene models in the reference genome, over 1/3 showed presence-absence variation in the diversity panel. And when we look at particular racial groups, we found specific paths differentiating racial groups. We've started to investigate the types and function of the genes exhibiting this presence-absence variation. And as a brief example, I'll focus on a gene class involved in defense response, specifically plant disease resistant genes. And we found that resistance genes were significantly more likely to exhibit presence-absence variation than we'd expect on average. And that this can be further evaluated both at the macro level, looking at the total number of resistance genes per racial group on average, and also at specific loci. For example, this RP1D, which is a major rust resistance gene in sorghum, and which you can see from this graph has large variation in frequency amongst the different racial groups. Okay, so moving on and back to our data resources panel. Another data resource I'd like to show you is the Sorghum QTL Atlas. Again, this was a world first QTL resource that we developed for the global community. But first, let's step back and explain why there was a need for this type of resource. Well, over the last 2 decades, hundreds of QTL and GUS studies have been published, and sorghum produces huge amounts of information on quantitative traits, where they are in the genome, what's the size of their effect, are they interacting with other regions? But making use of this information is not straightforward. And why? Well, that's because there was no central repository of QTL studies that allowed users to make these comparisons against a common framework. Now, here I thought I needed to write a note to the early career researchers. They may not realize that until a decade ago, the vast majority of trait dissection papers in sorghum used biparental populations and genetic linkage maps and made no link to the reference genome. So that's a lot of work to do. And the reason why we didn't have that collated data set was because QTL studies are very heterogeneous. They're very variable in quality, meaning that significant time is required to integrate and generate a comprehensive QTL database, and it's just too exhausting to contemplate. But in 2009, David Jordan and I did contemplate it, and we published the first study that attempted to not only consolidate all of these papers, but also adjust for QTL confidence interval. That's the size of the QTL based on parameters in the original study. And at that point, and the reason that we didn't collapse through overwork was the database consisted of only 44 studies at that point. So we mined all of the QTL, which was less than 800, and aligned them to the first version of the sorghum genome assembly, as well as to a sorghum genetic consensus map that I had developed with David as well in 2009. That was all very well, but as soon as we published it, it was pretty much out of date. So we've continued to update this resource, and 10 years on, in 2019, we published an update. And as you can see, now we've got fourfold the number of studies. So from less than 50 to over 200, from about 700 QTLs to over 7,000 QTLs, and now we aligned them to multiple versions of the sorghum genome, still to the consensus genetic map, but also to maize and to rice. And this sort of resource can more readily help researchers address questions that were previously very time-consuming to figure out. And the one that I will focus on is which genes are within the QTL for my target trait in sorghum that are also in syntenic locations in maize and rice, to link in with the work that we're doing on the grass pangenome. So once again, this slide shows 10 chromosomes of sorghum going around in a clockwise direction. The next 3 lanes, B, C, and D, show the comparable or syntenous regions from rice and maize. So for any given QTL location in sorghum, we can predict the syntenic location in maize and rice. And linking to whole genome assembly resources for both species, we can identify all of the predicted gene models within the QTL-CI in both maize and rice. As an example, I've highlighted the location of the gene Phi-B within a QTL for maturity on sorghum's chromosome 1, and the predicted syntenic locations in rice and in maize, both of which also contain the Phi-B gene. And the atlas links to external resources for these species to further interrogate these genes. And in the atlas, we also make use of previously published resources that provide information on which genes are under selection, not only in sorghum from our original resequencing paper, but also in comparable studies in rice and in maize. And one of the main reasons that we've included this information is because it allows us to take advantage of known gene order conservation or synteny between closely related species, and the fact that there are many-fold more QTL and GWAS studies in rice and maize than there are in sorghum. And by using them, we can piggyback and identify genes that are in common across species and make use of further investment in maize and rice on functional characterization in other species. So back to the data resources panel, and on to the last point I briefly want to mention, that is genotypic, pedigree, and phenotypic data. And the main point I want to make about this is the growth in the volume of these data types in the last 10 years by our program. So obviously for genotypic data points, which we've been giving us increasingly higher coverage of the whole genome, but this is now being dwarfed by the growth of phenotypic data. And this is driven by the adoption of new platforms and sensors, including drones, resulting in hundreds of experiments with tens of millions of data points across a large number of traits. One point to highlight is the importance of using consistent data standards across experiments. So that is consistent genotype names, consistent trait names. And if you ensure concurrence or overlap of genotypes across years, then you can make use of advanced statistical models to increase accuracy. So what I have shown you here is a range of genetic populations and a range of data resources. The principle we apply to research is to make them reusable, interlinked, complementary. In this way, we can reduce the cost of research as we don't have to generate bespoke or custom populations for each new project that we start, and we can reuse historical data, which contributes to an increased pace of research and a reduced time to impact. So what I'd like to do now is briefly focus on the analysis and use of this data to address both a range of fundamental and applied research questions. And I'd like to start off describing how we approach the identification and prioritization of potentially useful traits to select for inbreeding, which starts with an understanding of the production environments and the identification of important complex traits. Based on our knowledge of trait biology and through trait modeling, we can identify candidate traits, which we can then use to search for genetic variation in our different populations. And once favorable variation has been identified, we can feed back this data into the model to help us predict the consequences of modifying that trait in particular genotype by management scenarios. I thought a real-world example would be helpful, and I'm going to focus on root angle. Graham Hammer's group, together with David Jordan, conducted a number of studies on nodal root angle in sorghum and observed that changes in the root angle are likely to influence water capture in different environments by modifying both the horizontal and the vertical exploration of the soil, and that modeling with best bet assumptions indicated that substantial yield gains in some environments were possible if correct root architecture was deployed in any individual management by environment context. So, knowing that there was a potential real benefit from this trait, we needed to know what variation existed in our germplasm. So a rapid screening methodology was developed by Vijaya Singh in her PhD and used to screen a diverse number of lines from the parents of our interlinked populations. And this initial screen revealed considerable variation. And having found significant variation, a more high throughput screening platform was developed and used to screen our multiple populations. And this work was conducted across two specific projects. The first, a joint Gates project, an ACR project led by David Jordan and Graham Hammer, and the second, a joint ACR IRDC project led by Andy Borrell. So with Vijaya's system, we screened and analyzed all of our populations using that platform. We analyzed the data and identified QTL in each population separately, and then in comparison with one another, and then integrated the output with our QTL atlas, resequencing and pan-sequencing, pan-genome resources. But the more interesting element of this particular trait example that I want to highlight today is how the integrated nature of the resources allows us to access pre-existing phenotype data, including yield and stay green across the populations, and hence explore the association between nodal root angle and yield in more detail and entirely without generating more data to answer the question, does root angle make a difference? So using our database of historical data, we could identify the allelic state of genotypes and compare with pre-existing performance data across many years and many environments. And through this in silico study, we identified that the genetic regions controlling nodal root angle also had an effect on yield and stay green, and that yield was increased in the presence of narrow root angle in certain water-limited environments that typify the production area in Queensland. So in other words, data from pre-existing multi-environment trials told us that narrow root angle is associated with the stay green drought tolerance trait and with higher yield in most Australian sorghum growing environments. And other types of environments are, of course, sampled within our target population of environments, and crop simulation modeling allows us to predict the nodal root angle alleles which are most advantageous in particular environment by management situations. And this is an example of how crop simulation modeling can be viewed as a linking technology to address a number of what-if questions, including what root architecture QTL alleles would work best in my current management and environment systems? What is the likely risk associated with growing the best combination? Our power to answer such questions is also enhanced by using genomics as a linking technology, which can also enable us to answer a range of questions, such as, are the genes for root architecture segregating in my breeding program? Does variation in these genes have different effects in different genetic backgrounds? Such an integrated systems approach to complex trait dissection using a set of interlinked resources provides many advantages rather than focusing on one discipline in isolation, and has allowed us to identify that large variation exists and that we could see that it has a temporal and spatial pattern in its water use. We have identified that it was linked to grain yield under drought, and we could rapidly identify genomic regions and alleles that varied significantly for nodal root angle across populations and species. And we can also have the potential to deliver to industry rapidly through the use of accessible and relevant genetics. I'd like to give one more trait-specific example for the application element of my talk. This one is focused on grain size. This work arose from an ARC discovery project together with Ian Godwin, David Jordan, Jimmy Batella, and Yong Fu. So to give you some background, grain size is a key yield component of cereal crops and a major quality attribute, both for grain growers and for grain users. And what do processors want? They want bigger grain and they want consistent grain size. And what do growers want? They want the same yield, but they do not want to compromise. They do not want that yield overall to be compromised. So why would it be compromised? Well, there is a trade-off between grain number and grain size, where as the grain size increases, the grain number decreases. However, variation in grain weight independent of number still exists. But this trade-off is complex, and to access this slice of variability requires understanding the different factors contributing to grain size. So grain size in cereals is a function of both the potential maximum grain size and the capacity of the plant to fill the grain. While the potential maximum grain size is largely genetically predetermined, the capacity to fill the grain is strongly affected by a similar availability. So we developed a phenotyping treatment to try to separate the capacity to fill the grain from the potential grain size by removing half of one of a pair of selected heads within a plot to remove a similar availability as a constraint. Yong Fu applied this phenotyping treatment to both the diversity panel and the NAM resource that I described earlier, and then developed a processing pipeline to measure a number of grain attributes on thousands of harvested heads from both populations. It's a huge investment of time. Unfortunately for Yong Fu, just after he finished the project, we invested in a new seed counter, which was not only significantly faster, but also able to measure vastly different features of the samples, which is pretty bad news for Yong Fu. But he was a very resilient chap and he powered on regardless, and he generated excellent phenotypic data. He integrated it with our pre-existing genotypic data and mapped multiple grain size measurements across the two treatments and across both populations, and identified 50 QTLs across populations and treatments, with HH denoting the QTL identified in the half head treatment and FH, the QTL in the full head treatment. And by comparing across treatments, 21 QTL were detected only in half heads, which are more likely to be associated with inherent genetic potential of grain size that is affected by source availability, whereas 18 QTL were identified in full heads and are more likely to be associated with the capacity to fill grain. With the identified QTL, we could then integrate with our other genomics resources to identify candidate genes. I've shown one of the candidate genes here, GS3. a well-known gene controlling grain size in rice, which is highly expressed in sorghum early inflorescence, and which we found to be under selection during sorghum domestication. GS3 increases grain size, but appears to have little or no impact on grain number. So to summarize this overall approach for this particular example, physiological understanding of the trait enabled us to identify a method to expose the two different drivers of grain size, the potential maximum grain size and the capacity of the plant to fill the grain, and to apply this to pre-existing and already genotyped genetic populations, and then to integrate the results with other pre-existing data resources to identify targets for breeding and gene editing. So to summarize, I've given a brief overview of the resources and the tools we've developed with a major focus on the genomics resources and how we use them in a range of research projects. I gave you two trait-based examples and how we've used these interlinked resources, but I wanted to highlight here the range of traits we are working on currently or have in the past using these approaches. And to finish off, I want to again highlight that the real power comes when you employ an integrated approach to build bridges across scales and disciplines, to understand context dependencies and operate in relevant genetic contexts. We focus on exploring natural genetic diversity, but we ensure that we work at sufficient scale for the problem. And I hope that I've demonstrated, we maximize interdisciplinary research and data sharing between groups. So once again, I'd like to stress that the work I've described is multidisciplinary, and I'm very fortunate to work with an amazing team. And here I would like to acknowledge and thank the team in Queensland spanning both DPI and UQ with genetics work led by David Jordan, who has an amazing vision to integrate islands of knowledge across disciplines and harness this for sorghum genetics and genomics. It is a real privilege to work with this team. I'd also like to acknowledge collaborators nationally and internationally. And of course, I'd like to acknowledge and thank funders, including the Queensland Government through DPI, the ARC, ACR, the Crop Diversity Trust, Gates Foundation, IDRC, and GRDC. And finally, thank you for listening. And for your enjoyment, I created images of a field of sorghum in the style of three of my favorite painters, Renoir, Picasso, and Van Gogh, with the help of my mate ChatGPT, who helped me create a few of the images I used in today's presentation. So thank you.
Thanks so much, Emma. Fantastic talk. We've got time now for some questions, and we might take a mixture from the floor and some from the Q&A.
Thank you for your talk. I have two quick questions. One is hopefully not too simple. The first is, what is a gene in this context, actually? Because I know that hopefully that's not too simple of a question.
No, it's a great question. So a gene in the context of a pan-genome is one that has sequence features that define it as a gene. So it will have a starting codon and an ending codon. So it is a sequence-based prediction algorithm that identifies it as a gene. On top of that, one can overlay whether or not it's expressed. And our process and pipeline that we used in our pan-genome development did include that element as well.
And then the second question is, do you think that those core genes are sort of the evolutionarily established genes throughout, the history of even thinking hundreds of millions of years ago as life was developing, and that those cloud and shell genes are really just key things that are being toyed with kind of by evolutionary selection, kind of like drift with alleles almost what those are undergoing a selection trial almost to see if they're useful?
Well, certainly the function classification of the core genes compared to the dispensable genes certainly indicate they've got a lot more core functionality and basic biological processes. But it will be interesting to compare the core genes from the 3 species pangenome compared to the sorghum core genes in the sorghum pangenome, and to see how many similarity and differences there are. I think the dispensable genes within the sorghum pangenome, we could see that there was a lot of genes there, for example, that enabled sorghum to adapt. And one of these was related to flowering time, where depending on whereabouts sorghum is and what latitude or longitude it's at, it needs to be able to move around and maintain diversity to keep its evolutionary advantage. So for the sorghum pangenome, our interrogation of this dispensable genes certainly indicated a more adaptive opportunity. How this relates to when we do the 3 species comparison, I'm looking forward to looking at that.
Yeah, I almost wonder if they'll become core genes eventually, and then lead to speciation rules, essentially a different environment.
Yep.
Okay, we'll take a question from the Q&A. We've got a question here from Angel Popper. Have you found trait-specific benefits of the newly identified structural variants?
Have we found trait-specific benefits from the newly identified structural variation? So we certainly looked at that first of all, and we found that there were some versions of grain color that have a specific presence-absence element to them. We also know that one of the key genes that is involved in resistance to a parasitic weed in Africa called striga is linked to presence-absence variation. Those are 2 immediate examples that spring to mind because they had very striking large structural variations in the genes themselves. But I think the answer is we will potentially find a lot more, but the challenge will be when we can identify, based on LD, whether or not it is the actual structural variant that is the causal mechanism, or is it a SNP in LD? And that is the sort of thing we're starting to grapple with if we want to start to use presence-absence markers in a GWAS or mapping approach, where really you might be looking at a presence-absence marker and you think that that's your causal, but depending on the LD in your population and what's going on around, you may not actually be looking at the causal. But for sequencing and those 2 genes, we have got definitive evidence that it was the structural variation that is creating this beneficial phenotypic response.
Well, thanks, Emma. And then a related question, I suppose, here is from Angel as well, is how does accurate genome annotation impact pan-genome assembly and the quality of the genome?
Yeah. Hugely, Angel, hugely. And you will be able to get many different annotations with every algorithm that you use to annotate. So a common approach now is to use multiple algorithms and create a meta algorithm, if you like, or meta annotation, because every tweak, every parameter can create a different output. So I guess a note to younger scientists starting out is to be very mindful of the default parameters of any algorithm you're using. And sure, start with the default, but then it's always a good idea to just, for your own knowledge and understanding, change a few, look at the output, change it again, and start to get a feeling for when things change, and not just trusting that the starting parameters are going to suit your species and your data set.
Great, thanks, Emma. A good question here from Charles Ologidi. He says, great talk. How can this pattern of study be applied to underutilized species? So I guess referring to the integration of your research program.
Well, I guess this is where comparative genomics really comes in. And because so much sequencing has been done now that depending on the clade or the taxonomic lineage, the particular under orphaned crop is in, it is quite likely that somewhere in that clade, somebody will have invested more heavily in resources and sequencing. So an example for sorghum is a native grass species in Australia called kangaroo grass or femida. And they're just working on that at the moment. And luckily is very closely aligned to sorghum at a genome synteny level. So kangaroo grass, which would not have any investment from GRDC, ARC, the big funding bodies, is able to leapfrog by using sorghum as a bit of a backbone for making some fundamental discoveries at the start. So I think comparative genomics is the initial way forward to start to demonstrate something and then build a more compelling argument at some point for additional funding. You can start cheap, but at some point you do need some money for genotyping larger populations and this sort of thing.
Another question here on the Q&A from Prema Kumar. Can we use Pan-Genome for QTL-seq or BSR-seq studies? And I suppose that raises the question, more broadly, can we get more informative about the function of these sequences, I suppose?
Yeah, and that's a really good question. And it's a question that has been challenging us for a while because all of our GWAS studies currently, in most of the common algorithms used, PharmCPU or Blink, assume a single reference genome. So if you've got a few methods, you can, of course, use multiple different reference genomes and align all of your sequencing to multiple different reference genomes one by one. Clunky, but possible, and make comparisons of scores that appear or disappear. But there are also new approaches, more KMA haplotype approaches that are coming up that enable you to sort of take the universe of haplotypes that you get from all of your de novos and look at them at one single time. Those are still not so commonplace yet. This is evolving sort of in real time, but I think that will be where we go, because right now it's a bit clunky. We've starting to get the knowledge, but at the moment our approach has been still do it against a single genome, but then go to our pan-genome in that area to discover what is happening around.
Okay, maybe I've got a question here on the Q&A from Shella Shaukat. What role do linking technologies like phenomics, genomics, modeling play in bridging scales from gene to field performance? Perhaps you could elaborate, Emma, on maybe sorghum. A sorghum gene is a good example there, perhaps.
Well,
I say they're all sorghum genes. A stay green gene is what I was thinking.
okay.
You might have another great example. Yeah.
It's a really good question, and linking technologies such as crop simulation modeling can be so powerful. And I really like the example that we gave with the nodal root angle, because it enabled us to enables us to better understand the environments where sorghum is grown, for one. So the real beauty of understanding that the same location doesn't experience the same environment from one year to the other helps you interpret your understanding of the outcome. So performance for yield, for senescence can be interpreted with the understanding of the particular environment type that was experienced, which does not remain the same in a particular location. So we're able to see water usage and whether or not water usage is a terminal drought stress or a relieved drought stress in a particular context, and understand how genes react relative to that environment type. It was quite a revelation to me to understand that different environment types occur at different frequencies at the same location. And the assumption was, if I'm doing a trial at Dolby, I'll be getting the Dolby environment. You'll be getting the Dolby environment in that year. And even then, depending on when your genotypes flower and mature, each genotype actually experiences its own micro unique environment, depending on when it matures and when it gets water relative to flowering. So modeling is an amazing linking technology that can really expand how you're considering the plant perspective.
Thanks, Emma. And I want to just ask one last question. You spoke about lots of linking technologies, and I'm stealing this from the Q&A as well a little bit here. What about gene editing? Is there a role for gene editing as one of these technologies in the toolkit?
Yeah, absolutely. It's a very exciting technology, and I did pay lip service to it just with the grain size gene that I mentioned. I think it's a very exciting technology. We would love to have it as part of our portfolio more readily. I think it's still a little bit expensive. It still has some bottlenecks that make it less high throughput at the moment, but we've got our eye on it, and we're talking and working very closely within Godwin, and certainly excited about the opportunities in the future, both for research, but also for deployment.
Great. Thanks so much, Emma. I think we've run out of time. So I'd like to thank Emma again for a fantastic seminar. Thank you. I would like to highlight our next seminar in the series. Will be on the 3rd of December. So please register and book up your calendar for this one. It'll be a great seminar from Dr. Jody Neal, who breeds strawberries. And I think, this is a fantastic breeding program, adopting a lot of technology and doing some exciting things. So you'll have to tune in to the seminar to find out. And of course, if you missed any of the seminars, please check them out online. They're all recorded. And if you want to relive Emma's seminar again, you can go online and check it out. So there's a lot of detail in these seminars, and often it takes a couple of times to really absorb some of that information. So that's what it's there for. Thanks again for coming. Thank you for joining online, everyone. And for those here in person, please help yourself to a tea or coffee and some snacks. Thank you.